Sawadee-krap. Good afternoon, everyone. Welcome to Tokyo Tech Research Showcase. I am Tatsuya Mizukoshi, the director of Tokyo Tech ANNEX Bangkok. I will act as facilitator for today. Before starting today's session, let me give you a few notes. First, about Q&A. When you have questions, please use the Q&A button in the Zoom toolbar, and you can post your questions during the presentation. The speaker will respond later in the session. We may not be able to answer all of your questions during this event. In such a case, please ask again in the questionnaire. Second, about questionnaire. A questionnaire will be sent to each participant after this event. It should take less than 5 minutes to answer. And please answer within 1 week by September 22nd. Your cooperation will be greatly appreciated. Today's presentation materials will be made available to those who complete the questionnaire. We will send the link for today's materials together with the questionnaire, and the password will be sent later. This event is co-sponsored by Tokyo Tech and NSTDA, and we have video messages from both co-sponsors as the opening remarks. First, Dr. Tetsuya Mizumoto, the Executive Vice President for Education of Tokyo Tech, will say a few words.
Thank you for joining us for the 2020 Tokyo Tech Research Showcase. I am Tetsuya Mizumoto, Tokyo Tech Executive Vice President for Education. Starting in 2007, with the development of TICE Tokyo Tech, an international joint graduate program, Tokyo Tech and NSTDA have built a positive relationship for the development of science and technology in Thailand and Japan. Since Tokyo Tech established Tokyo Tech ANNEX Bangkok in 2018 in Thailand Science Park, the relationship between Tokyo Tech and NSTDA has become even stronger. Due to the spread of COVID-19, many activities throughout the world, including economic activities, have been halted. In response to this, building on our capabilities in research and academia industry collaboration, Tokyo Tech has launched a series of special projects under the Social Rebooting Technology Initiative, which aims to counteract COVID-19 and bring about a stable and sustainable society. NSTDA has agreed to cooperate with Tokyo Tech in this initiative. We believe our collaboration will offer encouragement and results to the people of both our nations. An annual event of Tokyo Tech ANNEX, the Tokyo Tech Research Showcase, was originally scheduled to be held last March. However, we were unable to hold it due to COVID-19. I am very pleased today that through this online research showcase, we are able to introduce Tokyo Tech cutting-edge technology. The topic of today's research showcase is biotechnology for industrial use. As you know, agriculture and biotechnology is designated one of the key industries under Thailand 4.0. I believe this area is an important one, as it offers great potential for collaborations between Thailand and Japan. I have heard that many of you who registered for today's event are from research institutions, universities, and industries. I hope you will participate actively in today's event. If you are interested in exploring possible collaboration with Tokyo Tech, I invite you to contact Tokyo Tech ANNEX Bangkok. And finally, I would like to express our deepest gratitude to NSTDA, our co-host of the 2020 Tokyo Tech Research Showcase. And I sincerely hope each of you gets the most out of this special event. Thank you very much.
Thank you, Dr. Mizumoto. Next, greeting from Dr. Chadamatsu Tuwaseta-kun, the Executive Vice President of NSTDA.
Good afternoon, Professor Tetsuya Mizumoto, distinguished participants. Welcome all of you to the 2020 Tokyo Tech Research Showcase today. It is a great honor for NSTDA and for me to be part of this special event. As Professor Mizumoto said, NSTDA and Tokyo Tech has developed a close relationship through the TIE's Tokyo Tech program, which has been operating successfully for more than 10 years now. The program is a tripartite partnership between NSTDA, Tokyo Tech, and a number of Thai universities, aiming to establish an institution for human resource development to produce world-class researchers and high-level engineers at a master's degree level. And 2 years ago, in 2018, we have decided to extend our collaboration from mainly educational activities to research collaboration by using network of Thai's Tokyo Tech to accelerate academia industry research collaboration in Thailand, which are implemented under the umbrella of Tokyo Tech ANNEX Bangkok. We aim to collaborate in research areas that correspond to the industrial or societal needs of both countries. Such move correspond to the global trend where international collaboration is increasingly synonymous with research excellence, and NSTDA is very pleased to work with Tokyo Tech in this movement. As for today's event, the Tokyo Tech Research Showcase, which is an annual event of the Tokyo Tech ANNEX Bangkok, we have organized in a new normal way using an online platform due to COVID-19 pandemic. Thanks to the advanced technology, we are able to meet, share, and exchange knowledge despite physically being apart. The theme for today is biotechnology for industrial use. This theme is very relevant to the Thailand context because our government has recently adopted the BCG, which stands for Bio, Circular, and Green Economy, as a new economic model for inclusive and sustainable development. The BCG economy capitalizes our country's strength in biological diversity and cultural richness, and employ technology and innovation to transform Thailand into a value-based and innovation-driven economy. Furthermore, it conforms with the UN Sustainable Development Goals and is aligned with the sufficiency economy philosophy, which is also a key principle of Thailand's economic and social development. Thailand has developed a network of organizations that support research and development in biotechnology. Currently, 24 universities across the country have the combined capacity to supply approximately 7,000 students in biotechnology background each year. In addition, well-established research infrastructure, including various pilot plants located in several leading universities across the country and also in NSTDA, allowing biotech companies to scale up their research before commercializing their innovations. We hope to welcome a visit from our guest speakers and friends from Tokyo Tech, as well as industry from Japan, to these facilities in Thailand when our borders are open. But today, we will learn about some research in this area in Japan from our distinguished speakers. I wish the showcase today will urge all parties, whether industry, university, government agency, and related organization, to further pursue their interests beyond today's event, to discuss and develop strong collaboration among us in the near future. Last but not least, I would like to take this opportunity to thank all participants in this event today, and particularly our partner, Tokyo Tech, for your great effort and contribution to Thailand and our neighboring countries. Thank you.
Thank you, Dr. Chadamas. Then I welcome the first speaker. The first speaker is Professor Toshiaki Fukui. He belongs to School of Life Science and Technology. Today's title is Metabolic Engineering for Microbial Synthesis of Biodegradable Polyesters from Biomass Resources. Then Professor Fukui, please.
Okay, good afternoon, ladies and gentlemen. So, under the present situation, it is very difficult for us to visit the Bangkok. But today, I thank organizing committee in NSTDA and the Tokyo Tech ANNEX to give me an opportunity to talk about our research work in this great online event. Okay, so I would like to share the presentation slide. Okay, so today I would like to talk about the metabolic engineering for microbial synthesis of biodegradable polyester from biomass resources. So the brief background. So as you well know, plastics are now essential materials in our modern society, but the waste plastics are causing environmental problems. So most plastics are produced from petroleum, so incineration of waste plastics increases atmospheric carbon dioxide. So of course, petroleum will be exhausted in future. So, and most petroleum-based plastics have low degradability, very hard to be degraded. So, which leads to the plastic pollution of natural environment and exhaustion of landfill sites. Moreover, recent studies have indicated that the plastic pollution in marine environment is a quite serious problem. About 8 million tons of waste plastics are entered from land into the ocean per year. And these waste plastics are broken to small plastics called microplastics. And such microplastics are accumulated in natural environment as well as in biological systems. So we have to develop more eco-friendly plastic materials. There are two concepts for such eco-friendly plastics. One is biodegradable plastic. They can be used as well as the general plastic, but after the utilization, they can be degraded to carbon dioxide and water by environmental microorganisms. Another concept is bio-based plastics. So these plastics are produced from biomass, such as cellulose, starch, or plant oil, which can be regenerated by plants via photosynthesis and fixation of atmospheric carbon dioxide. So based on these two concepts, so this table shows a classification of some kinds of plastics. Most plastics are petroleum-based, non-biodegradable, and some polyesters petroleum-based biodegradable plastics such as polycaprolactone or polyvinyl alcohol. So recently, biopolyethylene and biopolypropylene are attracted as bio-based non-degradable plastics. But apparently, plastic materials with both properties, that is bio-based biodegradable plastics, are more favorable for the environment. So this class contains polylactic acid, and polylactic acid is produced by a chemical polymerization of lactic acid produced by fermentation. So we are now focusing on another material in this class, polyhydroxyalkanate, directly produced from biomass by bacterial strains. So it has been known that the various kinds of microbes accumulate polyesters of 3-hydroxyalkanate, termed polyhydroxyalkanate PHAs, as carbon and energy storage materials under unbalanced growth conditions. PHAs are bio-based plastics produced from renewable biomass resources, and PHAs are thermoplastics. So once after extracted from the cells, they can be fabricated into films or fibers like general petroleum-based plastics. Moreover, PHAs are biodegradable plastics that can be degraded by environmental microorganisms after the utilization. Thus, PHAs have been attracted as eco-friendly polymeric materials. The most frequently observed PHA is a homopolyester of 3-hydroxybutyrate P3HB. And one kind of bacteria, Ralstonia eutropha, is a famous P3HB producer, which can accumulate P3HB homopolymer up to the range from 60 to 80% from various carbon sources. The P3HB biosynthesis pathway in this bacterium has been well studied. That is, that 2 molecules of acetyl-CoA dimerized and reduced to R isomer of 3-hydroxybutyl-CoA, and then are polymerized by the polymerizing enzyme PHA synthase. The genes of these 3 enzymes, PHA synthase, 3-ketothiolase, and NADPH-dependent reductase are organized as the far CAB operon on the chromosome in this bacterium. So unfortunately, the physical and the mechanical properties of P3HB are not so good. So it is a very hard plastic and high crystallinity, similar to polypropylene. But the elongation to break is much smaller than those of the general plastic. So that is, P3HB is quite stiff and brittle material. So its wide application is very difficult. For improvement of physical and mechanical properties of bacterial PHAs, biosynthesis of random copolyesters consisting of 3HB and the second or third hydroxyalkanate unit has been attracted, because these copolyesters show the altered properties depending on structure and composition of the second or third hydroxy acid unit. So in the PHA producing cells, so biomass are incorporated into the cells and then metabolized through the metabolic pathway within the cells. So conversion, so biomass are incorporated and converted to the monomer units, and then polymerized, enzymatically polymerized to the polyesters. So our concept is the engineering of the PHA producing cells by genetic engineering. So modification of the biomass uptake and the metabolic pathway to form the 3-hydroxyactyl-CoA monomers, and also engineering of the polymerizing enzyme, PHA synthase. So these modifications are expected to achieve the biosynthesis of PHA copolymers from the desired biomass resources. So one example is this kind of the copolyester 3-hydroxybutyrate called a 3-hydroxyhexanate, having the C6 monomer unit. So about 25 years ago, a Japanese company, Kaneka, collaborated collaborated with Riken Institute, carried out a screening of microorganisms that can grow on and accumulate polyesters from renewable plant oils. So this effort resulted in isolation of a new PHA producer, Aeromonas caviae, from Japanese soil. So this bacterium can synthesize this random copolyester P3HB called 3HHX from long-chain fatty acids or plant oils as a carbon sources. So the introduction of the C6 unit, 3HHX unit into the polymer chain decreasing the melting temperature and greatly improves the flexibility. So this copolyester containing about 11 mole percent 3HHX fraction shows a 400% elongation in break, which is much larger than that of the P3HB homopolymer. So this copolyester is very flexible, suitable for the practical applications. We then carried out the genetic analysis of Aeromonas caviae and cloned the PAR-C PHA synthase gene with a broad substrate specificity to 3HA-CoA from C4 to C7 chain length. And we also found that the PAR-J gene located just downstream of the PAR-C encodes a new enzyme R-specific NOL-CoA hydratase. So this enzyme can catalyze the hydration of double bonds in trans 2-NOL-CoA of C4 to C6 with strict R stereo selectivity. So phenylacyl moieties are degraded by beta oxidation cycle in caviae. So 2-NOL-CoA intermediates of C4 and C6 are intercepted to form the corresponding R3-hydroxyacid-CoA by the function of the new enzyme PAR-J. They are accepted as a substrate for PHA synthase in caviae, leading to the synthesis of P3HB co-3HHX copolymers. So this was the first identification of the metabolic linkage between beta oxidation and PHA biosynthesis pathway. A flexible copolyester with 20 mole percent 3HHX fraction 3HHX composition was produced from vegetable oil by Aeromonas caviae, but unfortunately, the productivity was very low. So as I talked already, the R. eutropha can well grow. R. eutropha is an excellent P3HB producer. So this bacterium can well grow not only on sugars but also on vegetable oils. Vegetable oils. But R. eutropha accumulated only P3HB homopolymer from vegetable oil. But the production was more efficient than A. caviae. So we then start to engineer the R. eutropha or P3HB called 3HHX copolymer synthesis from vegetable oil. So as a result, we modified several metabolic steps in this bacterium, such as the deletion of the phi-A ketothiolase and also inactivation of the beta oxidation enzyme. Also, as well as the insertion of the two kinds of the phi-J R-specific hydratase. Then, as a result, the engineered strain can efficiently produce this copolyester containing about 12 mole percent 3HHX efficiently from the soybean oil as a carbon source. So the cells accumulated the huge amount of the polyester with the cells are shown in this electron micrograph. So this copolyester PHBH has been produced industrially in Japan by a Kaneka company from vegetable oil by using another recombinant strain of R. eutropha. the annual production was increased to 5,000 tons per year last year. So recently, many biodegradable plastics, including polylactic acid, are not so well degraded in marine environment due to the less bacterial density as a soil and compost environment. But the quite notable property of PHBH bacterial polyester is high biodegradability, even in marine environments. So PHBH is now attracted increasing attention as a possible plastic that can solve plastic pollution in the ocean. Okay, so we further continue this study engineering of R-Ultrafar to produce this copolyester, not only from vegetable oils, but also the sugars as a carbon sources. Because so the use of the abundant and inexpensive sugars as carbon sources is expected to be another possible way for the low-cost production. So in addition, this is challenging metabolic engineering because no wild bacteria capable of synthesizing PHBH efficiently from sugars has been reported so far. So R-Ultrafar wild strain H16 derived strains accumulated only P3HB homopolymer, even when the PHB synthase with broad substrate specificity is functional within the cells. So this was obviously due to the lack of pathway for supplying the C6 monomer R3HH-CoA. Another drawback of the H16 derived strain is a limited range of utilizable sugars. This wild strain H16 well grows on fructose and gluconate, but cannot utilize glucose as a sole carbon source. So this lack of the glucose assimilation ability is due to the lack of the uptake and phosphorylation of glucose moieties in H16 cells. So we have demonstrated that the point mutation in a component of gluconac-specific phosphotransferase system and the deletion of the putative repressor of NAGR allows the cells to utilize the glucose. So we have established the glucose utilizable strain of R-Ultrafar. We then designed the artificial pathway for P3HB co-3HX biosynthesis. So in this pathway, so the key reaction step is the formation of butyl-CoA as a precursor for further elongation to C6 and subsequent conversion to R3HX-CoA within the cell. The butyl-CoA is produced by reduction of double bond in crotonyl-CoA by the function of CCR. So however, so this enzyme CCR is bifunctional crotonyl-CoA carboxylase and reductase catalyzing not only the reduction of crotonyl-CoA, but also catalyze the reductive carboxylation to form the acyl-malonyl-CoA from crotonyl-CoA as a common substrate. So this formation of acyl-malonyl-CoA is the byproduct pathway. So we just utilize mammalian acyl-malonyl-CoA decarboxylase called EMD in order to convert back the acyl-malonyl-CoA to butyl-CoA. The resulting engineered strain produced PHBH, PHB-CoA-3HX from glucose as a sole carbon source with high cellular content of 80 weight %, but the 3HX composition was only 2.3 mol % due to the efficient conversion of acetyl-CoA to R3HB-CoA. So this low CHH composition is not so good for the practical applications. So we have engineered to aiming to increase this CHH composition. So when this FAB1 gene was deleted, the R-specific reduction mediated only by the minor FABC enzyme. So as a result, the C6 composition was actually increased, but the increase. So that this increase of the C6 unit by the FAB1 deletion strongly suggested that the protonil-CoA, which is a precursor of the C6 monomer, is generated through S3HB-CoA derived mediated pathway. So this flux was supposed to be relatively increased when FAB1 was absent. So the composition was highly increased, but the total production was decreased, unfortunately. So then we engineered the strain to enhance this S3HB-CoA derived pathway. So in this cells, these two reaction steps are mediated by unknown enzymes. So we started to identify these enzymes in R-Eutropha. So then we identified two kinds of enzymes, acetyl-3-HB-CoA dehydrogenase, and also new crotonase enzyme. So we then inserted the two genes into the chromosome of R-Eutropha. Then to enhance the reverse beta oxidation cycles forming protonil-CoA from acetyl-acetyl-CoA via the S-specific pathway. And then the broad substrate specificity of HAD and CRP2 were expected to be useful for conversion of both C4 and C6 intermediates. So the MCL, so further, the MCL specific R hydratase J4A derived from R-Eutropha was further replaced to the shorter chain length specific R hydratase derived from A carrier. So these modifications increased the C4 unit with maintaining the high amount of high composition of the C6 unit, resulting in production of this THBH copolyester composed of 12 mole percent CHH unit from glucose as a sole carbon source. And the pH content, cellular content was also quite increased, enhanced, so leading to the 75% dry cell weight. So this 12 mole percent composition of CHHX was very adequate. This copolyester shows adequate flexibility suitable for practical applications. So we expected this the engineered strain is useful to produce this copolyester from glucose or sugars as carbon sources. Okay, so the next is the last topic of my talk. This is PHA production from carbon dioxide. So this Ralstonia eutropha is also known as a kind of hydrogen oxidizing chemolith autotrophic bacterium. So this strain can well grow and synthesize the cis-HB homopolymer also on mixed gases of carbon dioxide, oxygen, and hydrogen as an energy source. And the carbon fixation is mediated by Carlin-Benson-Basham CBB cycle in these cells. So we can carry out this autotrophic flask cultivation of this bacterium by putting the mixed gases into the headspace and repeated exchange of the substrate gases. But this flask cultivation, so the large-scale cultivation is very difficult by this such the flask cultivation. So my collaborator, Professor Tanaka in Kinzai University, Japan, has investigated the P3HB production from carbon dioxide with hydrogen by Algal. As you know, hydrogen is flammable, so the very dangerous when mixed with the oxygen. So then he established the recycled gas closed circuit culture system to maintain oxygen and hydrogen concentration lower than the explosion limit. So then he achieved the high efficient production of the P3HB homopolymer from carbon dioxide by this system. So we then further considered to the whether the production of the PHBH copolyester from carbon dioxide is possible or not by using the engineered Algal strain. So we found that this answer is yes. The engineered strain having the artificial metabolic pathway can synthesize the this copolyester efficiently from the carbon dioxide and hydrogen. So we are now further focusing the further efficient production or the regulation of the composition. Okay, the summary of this my talk, and I thank my laboratory members and in Tokyo Institute of Technology and my collaborator, Professor Kenji Tanaka. Thank you for your kind attention.
Okay, thank you very much. Then I check the question, maybe no questions from the audience. So let me ask some questions from my side. It is very interesting that the characteristic of the copolymer changes so dramatically. And what kind of application can we enjoy by this copolymer?
Okay. So this copolyester is very similar to polypropylene or high density polyethylene. So the wide application is possible, we are thinking.
But generally speaking, high degradability means susceptible to hydrolysis. I mean, easy to get hydrolysis. How about the durability if we use the copolymer actually?
Yeah, this is a very good question. So we hope to use this plastic with no degradation when just we are using it. But I expect a high degradability after the utilization. So the recent research is attracted is focusing on the switch of the degradability. So to confer the ability, to confer the switch ability to the polymer to start to initiate the degradation after the utilization by sensing the pH or temperature or the further physical factors, environmental factors.
Okay, and in the case of Ralstonia eutropha, can we select autotrophic condition or heterotrophic condition? Which method is better for industrial use?
So autotrophic growth condition requires supply of the hydrogen and along with the oxygen. So this is very difficult to supply safety. So we have to establish the good system, good instrument to supply the mixed gases. So, but the heterotrophic condition is very easy to carry out. And so when we get the inexpensive carbon resources such as starch or other biomass resources, the heterotrophic production is better. But in future, we have to consider to produce such the polyesters or other compounds from carbon dioxide. So I think this is a very important future technology.
Okay, and one question from the audience. For feedstock, like vegetable oil, could you tell how much the cost for commercialized production as alternative materials comparing with PLA?
PLA, lactic
Yes.
acid. Yeah. So actual production cost is very important, and so such the information has not been well opened from the company. But I think that the production cost and the price of PHA and PLA are comparable at present now.
Okay, thank you very much, Dr. Fukui. Time is up.
Okay, thank you for your attention.
So the next speaker is Professor Hiroyuki Ota. He belongs to School of Life Science and Technology, and today's title is Lipid Remodeling and Nutrient Starvation in Microalgae and its application for oil production. Professor Ota, please.
Thank you, Chair, for your introduction. I'm currently working on the photosynthetic organism that is different from the previous speaker, Professor Fukui. So in my case, today I talk about the lipid remodeling, some interesting system to adapt the nutrient starvation in microalgae and its application for oil production. First of all, I'd like to briefly introduce the forecast of why the biofuel should be produced from microalgae. This is a forecast of power unit for automobiles. This slide clearly indicates that even when the 2035, as you see in the right hand, just a moment, the power unit, like for plug-in hybrid or gasoline automobile, diesel automobile, for all of this, we need some liquid fuels because this automobile have an internal combustion engine inside. It reached at around 80%, more than 80,000, even 2035. This clearly indicates some liquid fuel will be continuously required in the future. And we should consider the reduction of CO2 emission from the aspect of well-to-foil. What is the meaning of the well-to-foil? It's from the production of the fuel to the usage of the fuel. And that is very important for the aspect of the CO2 emission reduction. And another importance for fuel oil, liquid fuel, is their energy density. This is a very clear slide to compare the energy density for several kinds of the power source like ethanol, LPG, natural gas. In the case of natural gas, the energy density is quite low, and when you compare to the liquid fuel, the liquid fuel are much higher at around 4-fold in this case. And to compare the compressed hydrogen, it reached 8-fold. And in the case of the lithium-ion battery, so you see that the energy density is more than 40-fold. This slide clearly indicates the energy density of liquid fuels so much higher than any other power sources. And another important thing is that when you consider the large-scale production rate among various biofuel production platforms, so any of the photosynthetic organism like chlorella and some cyanobacteria, and as you see in the left hand, so this is the Nanocropis cell. This is a strain of Nanocropis gaditana. If you compare to some land plant oil like palm oil, the production rate is more than 5-fold. It reached a very high amount. So this means that the usage of the algal oil is very important to consider liquid fuel energy. How we can manipulate the production of oil in some microalgae. Recently, several kinds of the nutrient-starved condition is considered to be very important for the usage. As you see here, minus nitrogen or minus phosphorus is an important condition to accumulate a high amount of oil. But when you compare two conditions, we are very much interested in the phosphate starvation because even in both cases, you see the very high accumulation of oil inside as a green color. But the if you see the chloroplast fluorescence, the strong fluorescence still remain only under the phosphate starved condition. That indicate the chloroplast function still remain under these conditions. That is very important to consider the growth of the cells. What happens under the phosphate starved condition? One of the important adaptation mechanisms is lipid remodeling in the cell membrane. When the microalgae suffer from the phosphate starvation, many of the phospholipids are degraded by this system and supply the inorganic phosphate in the cells. And instead, in the cell membrane, several kinds of the other polar lipid without phosphate are accumulated. Many of the microalgae contain the betaine lipid instead of the phospholipid that is useful for adaptation of the phosphate starved condition. This is a nice case for nanocropis cells. As you see here, under the phosphate starved condition, you see the strong decrease of the phospholipid. The major component of phospholipid is phosphatidylcholine, and on the contrary, you see the strong increase of the DGTS, betaine lipid, particularly for the extraplastic membrane. When you consider the plastic side, I mean the chloroplast fraction, only 4 membrane lipids are contained. That is MGDG, DGDG, SQDG, and only one phospholipid is PG. And of course, even in the case of the chloroplast membrane, you see the strong decrease of phosphatidylglycerol, but instead, you see the strong enhancement of the accumulation of sulfokinols. This is sulfur contained glycolipid, and this kind of the glycolipid can substitute the function of the phospholipid. This is a case of the phosphatidylcholine. So you see the structure of the phosphatidylcholine and DGTS. Beta lipid is quite similar. The phosphatidylcholine have the negative charge here, the phosphate molecules. But in the case of DGTS, you see the carboxyl residue is here. So this can replace the phosphatidylcholine function to some extent. This is another case inside of the chloroplast. When you see the decrease of the PG, so you can replace the PG function by SQDG accumulation. So the both SQDG and PG had a negative charge here and here. That's why, so the SQD can replace the function of PG inside of the chloroplast to some extent. By utilizing this kind of the very interesting specific mechanism for photosynthetic organisms, we can manipulate the oil accumulation inside of the cells. What we do is, firstly, we find the specific promoter for SQDG synthesis. I mean, the SQD2 are strongly enhanced specifically under the phosphate stable condition, as you see here. And so we can obtain some specific promoter from SQDG gene, SQD2 gene. And then, so we can connect this kind of the very specific promoter to tag biosynthesis genes. We firstly select the chlamydomonas tag biosynthesis gene. We particularly interested in the final step of tag biosynthesis. I mean the tag is biosynthesized from diacylglycerol to triacylglycerol by the function of the several kind of the DGAT genes. But most of the genes are strongly upregulated under the phosphate stable condition, but only in the case of DGTT4. Here you see the DGTT4, the activation of this gene is not so strong. So that's why we decided to use this promoter to connect to the DGTT4 genes. And then we observed the accumulation of oil droplet inside in the transformant. Here you see the very clear the activation and the accumulation of oil droplet only under the phosphate stable condition, but in the control, no such kind of the activation are observed. So then we decided to utilize this very useful mechanism to the more useful microalgae nanocropis, because the nanocropis has a higher oil content. And another important thing is fatty acid composition is very simple, and this fatty acid composition is suitable for biofuel production. Another thing is when you consider the cultivation of the nanocropis, the cultivation becomes a very high density. So that means very high oil can be produced by cultivation of these nanocropis cells. And when you want to manipulate the genes, we can use a homologous recombination for the manipulation. And genome sequence is now available for the NIS 2145 strains. This is currently very popular strain which can be manipulated by homologous recombination, and genome sequence is now available. And another important thing is high content of EPA. EPA is, so you know, the omega-3 fatty acid, another very useful fatty acid inside of the nanocrop cells. This is the case of the nanocrop cell. The situation is quite similar to the model alga chlamydomonas. When we put the minus nitrogen or minus phosphate condition, so you see the big decrease, big accumulation of the oil droplet as a fluorescence of the nylate. And when you analyze the inside of the structure, the only in the case of the phosphate substrate condition, the cells still have the large chloroplast. That means that they still can continue the growth, even under the nutrient-starved condition, with accumulating a high amount of oil. And after introducing this specific promoter set, we firstly check the expression of DGTT4 genes. And this is a result for any of the transformant. So you see the very specific induction of the DGTT4 gene. This is derived from the chlamydomonas, but in the nanocrop cells, the chlamydomonas gene are specifically upregulated only under the phosphate stable condition. This is quite nice system to manipulate the oil accumulation. And as a result, so you see only under the phosphate stable condition, of course, even in the wild type cells, you see the big increase of the polyacylglycerol content, but by the transformation, so you see the enhancement of the increase of the polyacylglycerol accumulation only under the phosphate stable condition. So from these results, these important points are suggested that some regulatory mechanism under the phosphate stable condition may be present in between a completely different microalgae. I mean the primary end symbiont like chlamydomonas and secondary end symbiont nanocrobes. Probably you know that for this kind of the microalgae, the origin of the microalgae, evolution of the microalgae are completely different. In the case of primary end symbiont, the origin is the same with the higher plant, land plant. But in the secondary end symbiont, the evolution history is completely different. Another host cell acquired chloroplast function by acquiring some eukaryotic photosynthetic organism. In the case of nanocrobes, red alga are suggested. And then they acquired chloroplast function. So the evolutionary histories, even evolutionary histories are completely different. Some regulatory mechanisms are highly conserved. Now we are interested in why this kind of the lipid remodeling system are so highly conserved. What is conserved in the mechanism? How the lipid remodeling regulated? Currently, we already know the major regulatory factors for phosphate starvation. Almost 10 years ago, one important regulatory factor are identified from chlamydomonas that is called PSR1. And then the important regulator are isolated from land plant that is called PHR1. This kind of regulator is now considered as a master regulator in both cells for the microalgae and land plant, and they have the important function in the upstream of the host cell stable condition. But we are now consider that some downstream regulator specifically regulate lipid remodeling may be present, and why we want to isolate the genes. And to isolate these genes with a co-expression database, firstly, we establish a AlcoDB database according to the success of the higher plant story. Now, many of the people consider the co-expression analysis a good way to identify some new important genes by utilizing some good marker. And this means generally in certain condition, some gene set which have the relative function are regulated simultaneously. If we analyze the co-expression, we can identify the new important target using the good marker gene. Okay, so this is a way to identify some good regulatory factors utilizing as a good marker. We firstly decided to use DGTT1. As you know, this is the final step of the tag biosynthesis, and DGTT1 is an important gene under any of the nutrient starved condition to synthesize polyacylglycerol. And generally, DGTT1 are strongly upregulated, and then under the nutrient starved condition in both nitrogen and phosphorus starvation, the tag accumulated very much. So we decided to use DGTT1 as a marker gene. And finally, by the co-expression analysis, we found several good targets. And among these targets, we finally selected a homologue of a higher plant MIB16.4 because if you see the host cell stable condition, the gene expression are strongly enhanced at around 10-fold. This MIB-like gene belongs to the R2R3 type of MIB protein family, and we further analyze the profile. Finally, we named this gene as lipid remodeling regulator 1 according to their function we identified later. And this slide clearly shows a strong correlation between DGTT1 and LRL1 are present. The correlation coefficient is more than 0.8. And another interesting is expression profile. So generally, the PSR1 is master regulator. This PSR1 are also highly responsive to the host cell stable condition, but this gene enhanced, strongly upregulated at the early stage. But in the case of LRL1, the activation profile, the upregulation profile is slightly different from the PSR1. This gene are rather upregulated in the later stage of the host cell stable condition. Then we identified two knockdown line for LRL1 protein gene. One is LRL11, other is LRL12. Please note the parental line is different for these mutant line. Even so, the phenotype is quite similar. Only under the host cell stable condition, you see the pale green phenotype here and here. Another interesting is the cell size and oil droplet accumulation. For both parental line, you see that under the host cell starvation, the cell becomes much larger according to the host cell starvation. But in the case of the mutant, the cell doesn't become so large. And another important thing is the accumulation of oil. accumulation of strongly suppressed only in the mutant. This is a quantification of the oil accumulation in both lines. So you see the tag accumulation becomes almost half, as you see here and here. And this is the case of the SQDG accumulation. In the host starvation, you see the increase of SQDG, but in the mutant, no increase was observed. This slide indicates the current our working hypothesis for the function of PSR1 and LRL1. The PSR1 is a master regulator and worked in the early stage, and they have the partner. One of the partner is another transcription factor BHLH2 and scaffold protein TTG1. And using the similar partner LRL1 function in the later stage and regulate the downstream genes. And we then try to identify the regulator for non-necropsy cells. And we already find several kinds of the homologue of PSR1. One is PSR1, that is a central regulator for the host cell condition. And we also identified homologues we call the PSR-like 1, PSR-like 2. Now, we already obtained a knockout for 2 genes, and when we analyze the phenotype, such kind of the strong accumulation of SQDG and DGTS, specifically under host starvation, are completely impaired in the mutant cells. This is the expression of the genes. The wild type cells, all genes are strongly upregulated, but in the case of the mutant, no such kind of the upregulation are observed. And by using these kind of the regulator, this regulator is very useful to manipulate oil production and by modulating their functions. And we currently have the good collaboration for the Hiroshima University and the Matsuda company to make the high oil producing microalgae utilizing the activation tagline. We already obtained several good candidates through the screening, and then we already established a genome editing technique for this non-necropsy cells using the platinum tolerance system. This is a model case. And finally, the Matsuda company evaluated the goodness for oil production by using our transformer. Another important thing for non-necropsy is accumulation of the omega-3 fatty acid. The location site is different. We can separate the utilization for oil production and more useful omega-3 fatty acid production for non-necropsy. Thank you very much for your attention. These are collaborators for me, and so Matsuda company is our good supporter for oil production. Now we have many other nice collaboration, and thank you very much for your attention.
Thank you very much, Professor Ota. Any questions from audience? Okay, one question from my side. Maybe for large-scale production, what kind of facilities are required? For example, raceway, fermentation, or something like that?
Yeah, in our case, we just started this kind of work using the test tube level. And we need to so large level of cultivation in the future. And of course, we consider the for oil production, for example, the use of the automobile, we needed the open pond.
Mhm. Okay, time is up. Thank you, Professor Ota.
Thank you very much for your attention.
And the next speaker is Professor Kiyohiko Nakasaki from School of Environment and Society. Today's title is New Biotechnology, Enhanced Utilization of Resources and Waste for Environment.
Okay. Thank you, Mr. Chairman. So first of all, I'd like to express my sincere thanks to the committee of this showcase for providing me a chance to have a presentation. So let's start my talk. This slide shows the title of my talk, The New Biotechnologies Enhance Utilization of Resources and Waste for Environment. This slide shows our research activities. The aim of our research is to solve environmental issues by microbial activities. The research involves waste reduction, energy and materials recovery. We are now treating mainly three topics. One of them is the biorefinery, in which we produce commodity chemicals from biomass materials. I show you here the bamboo powder, and we can convert it to the bioethanol. And our second topic is the biogas fermentation in order to produce energy from waste biomass materials. Biogas, as you know, the mixture of methane and hydrogen, and we can convert these gaseous fuels to the energy. And our third research topic is composting, in which we produce organic fertilizer from waste biomass materials. I can show you here the garbage, and so we can convert it to the compost. Today, okay, so next slide, yes. So we consider we are able to help construct sustainable society by combining cascade use of biomass and biomass reproduction. We start from the biomass, and we can produce many chemicals by the biorefinery. And this process emits the waste material. And we can convert it to the biogas by using the waste material from the biorefinery as the raw material for the production of the biogas. And we can obtain the energy, and then this process also emits the organic waste, and we can convert it to the compost. And after use of this material and energy, the carbon dioxide is generated again. But we don't need to be anxious about the increase of carbon dioxide in the atmosphere because the carbon dioxide is utilized for the reproduction of biomass by the action of the photosynthesis. So by combining the cascade use of the biomass and the biomass reproduction, we consider we will be able to help construct a sustainable society. Today, I'd like to focus on the composting. And this slide shows our new approach, DNA engineering. This word we advocate. So this word is a new word, and we advocate to use. The DNA engineering is very different from the genetic engineering. The DNA engineering is the DNA information-based engineering. By this, using this engineering, we can control the bioreactor with high performance based on DNA information. And also we can control the quality of biological product based on DNA information. So today, I'd like to show you one example of DNA engineering in the field of composting, making a compost. So this slide shows the production of the compost from the garbage, the kitchen refuse. And this is a composting process, composting time progress, we can obtain the compost. The compost looks like a soil, but it contains a nutrient, high concentration of nutrient for the plant growth. And the bottom picture indicates the plant growth assay. The left-hand side of this picture indicates the komatsuna, the Japanese spinach. Komatsuna grows when we apply the chemical fertilizer. And the center pot, no fertilizer, and the right pot indicate the komatsuna grows when we apply well-matured, good quality compost. And we observe the better growth of the komatsuna when we applied well-matured compost compared with the chemical fertilizer. So the researchers in this field and the manufacturers of the compost, they want to know how long does it take to obtain such good quality compost. So before we started our research, it has been long believed that the longer the composting period, the higher the quality of the compost becomes, like wines. But we found out that is not true, and that is just a superstition. In order to find out a good quality compost, the best way was to grow the plant. So the plant growth assay is the most reliable method, but it requires long time and much labor. So I would like to invent a new method which can determine the compost maturity with ease. So our new method for determining the compost maturity relies upon the molecular biological method. And this slide shows the relationship among the plant growth, the organic matter degradation during composting process, and the changes in microbial consortia. As you know, in the composting process, many microorganisms contribute to the degradation of organic matter, not pure cultured system. So they construct a kind of consortium. So, I'd like to explain the relationship among these three, and it is very well known that there is a strong relationship between the plant growth and the organic matter degradation. So we considered who degrade the organic matter. Of course, they are the microorganisms. So if we can relate the organic matter degradation and the changes in microbial consortia, we may be able to correlate the plant growth and the changes in microbial consortia. And if we can correlate these two, we can shorten the time to judge the compost maturity. When we use a plant growth assay, at least 6 weeks is required to judge the compost maturity. But if we can use the analysis of the microbial consortia, we can shorten the time considerably. This is our idea. So we started from this experiment. So we prepare the compost sample with different degree of organic matter degradation. So this is a composting reactor apparatus, and we put the garbage number 5 into the cylindrical reactor with a volume of 30 liter. And we put in the garbage into the reactor, and we then introduce the air from the bottom of this reactor. And the microorganism existing in the garbage consume the oxygen together with nutrients, and they emit the carbon dioxide. So we measure the concentration of carbon dioxide by the carbon dioxide analyzer continuously. And we inserted the thermocouple into the reactor, and we control the composting temperature at 60 degrees Celsius by regulating the air feed rate. So we want to know if we want to know the activity of the microorganism in the composting reactor, we need to multiply the air feed rate and the carbon dioxide concentration in the exhaust gas. And we can know the speed of organic matter degradation. And then we quantify the degree of organic matter degradation by calculating the conversion of carbon. This has a definition. The carbon loss as carbon dioxide until a certain composting time to the total carbon in the raw material multiply 100. This corresponds to the percentage of the organic matter degradation. And so this is the experimental result. And we conducted a composting experiment for 14 days. And so please look at this red curve, the conversion of carbon. The conversion of carbon started from 0%, of course, and then increased very rapidly in the earlier stage of the composting, then leveled off around 73%. So from this figure, we can say in the early stage of the composting, the organic matter degradation was very vigorous. We can know from this figure. And then we conducted a plant growth assay using the compost sample. So we conducted the plant growth assay. The method is advocated by MAF, the Ministry of Agriculture, Forestry and Fishery in Japan. And they recommend us to use the komatsuna, Japanese spinach. On the bottom figure, I already show you the time course of the conversion of carbon. And the red circles indicates a point where we obtained the compost samples. And then we carried out the plant growth assay by using these compost samples. And this is the photograph which shows the germination of komatsuna, and 0%, 20, and 40% correspond to the conversion of carbon. So we mixed the compost with 0% of organic matter degradation, that is, of course, the garbage itself, into the soil, and we sow the komatsuna seed and wait 4 days for the germination. And we can see here the growth of fungi, white color on the soil surface of the cultivation pot. The white fungi decreased significantly when the compost with 40% organic matter degradation was used, though the surface of the soil is still a little white. So if we use this compost, the pathogenic fungi may grow in the crop field. So we cannot recommend the farmers to use such dangerous compost. And I don't show you here today we conducted a similar experiment using the compost with 60% and 70 and 73% of organic matter degradation. And we didn't observe any white. So we can say those composts are very safe from the growth of the fungi. But we need to remember the 40% of organic matter degradation compost is still dangerous. This is very important. Please remember, 40% is still dangerous. Then we continued the plant growth assay until 6 weeks. And we took these photographs when we harvest the komatsuna. And the left lane corresponds to the komatsuna growth when we apply the chemical fertilizer, and the center lane we no fertilizer, and the right lane we mixed the compost with various degree of organic matter degradation, 0%, 60%, and 73%. And so, of course, the conversion of carbon corresponds to the composting time progress. And we are very surprised to hear the first, to observe first this result, because we observe the highest yield when we apply the 60% of organic matter degradation compost, not 73%. So I already mentioned when I started my presentation, the longer the composting periods, the higher the compost quality becomes. That is not true, and that is just a superstition I mentioned. And this is experimental proof. And then we so I show you the relationship between the conversion of carbon and the fresh weight of komatsuna gram per cultivation pot. Just from this figure, the compost with degree of organic matter degradation from 40% to 70%. These three composts are suitable for the application to the farmland. But please remember, I already mentioned 40% of organic matter degradation compost is immature and still dangerous. So by combining this result and our previous result, we can say only 60 and 70% of organic matter degradation compost is suitable for the application of the to the farmland. So next, we'd like to confirm, elucidate what happened in the microbial consortia. So we collected the compost sample and we applied the PCR DGG method. And we once recover the microbial cells and then follow this protocol, and we once obtain the purified DNA. And then we amplify the specific region of the DNA derived from the microorganism existing in the compost sample. And we once obtain the DNA, the mixture of the DNA fragment, and then we separate the DNA fragment by using the gel electrophoresis. And in our case, each band corresponds to each microorganism. And so I'd like to show you our result. This is a PCR DGG pattern. And so numbers from 0 to 14 above this picture indicates a composting time, day 0 to day 14. And we can see the bands, many bands. And so light intensity of the band corresponds to the concentration of that microorganism in the compost. And we noticed three characteristic bands. Band A so appeared from day 4, and existed at high cell concentration thereafter. And band B, this is so one of the characteristic bands, and was high concentration in the earlier stages of composting, then diminished gradually. And one more characteristic band, band C, they began to increase after the middle stages of the composting. So we so noticed these three characteristic bands, and then we compared the DGG pattern and the time course of conversion of carbon, and we noticed the succession from band B microorganism to band C microorganism. This timing corresponds well with conversion of carbon with the good effect to the growth of the plant, the most effective compost. So from the succession of these two microorganisms, we will be able to know the good quality compost. So then we next try to determine the name of these microorganisms. And this is one example. If we want to determine the name of this microorganism, we excise the DNA band and purify, then determine the DNA sequence 160 base, and we draw the phylogenetic tree. And from this tree, we know this band, the closest relative of Bacillus denitroficans. And so by applying the same method, we know the name of the characteristic band appeared during the composting process. And of course, the two important bands are the band B and band C, and band B correspond to Bacillus pallidus, and band C correspond to Bacillus samoglucosidaceus. So from the succession from Bacillus pallidus to Bacillus samoglucosidaceus, we know the best timing for the use of the compost when we expect the best growth of the plant. And the next slide is important. Please do not miss. I will use the animation here. The succession from band B microorganism to band C microorganism, we can judge the good quality compost. So we can determine the compost maturity by observing the succession of specific microorganisms, Bacillus pallidus and Bacillus samoglucosidaceus. Next, we try to invent more easier method. The principle is the same. So we try to invent the DNA probe, and which can detect the concentration of band B microorganism and band C microorganism. This is an example. If we want to determine the concentration of band B microorganism in the compost, we should design the oligonucleotide which can hybridize the band B microorganism gene with high specificity. And so if we succeed to produce this oligonucleotide, we attached the pigtail at the end of this oligonucleotide. And then this probe can hybridize with band B microorganism, and then at the tail we can expect the enzymatic reaction, and we can detect the existence of the band B microorganism by the blue color emission. So I show you the result of our experiment, and we extracted the band B, the DNA from band B microorganism, and we spotted on the filter paper here, and this is 33 femtomole, the amount, and we diluted successfully 1/3, 1/3, 1/3, and then 1/3. We diluted. And we observed the blue color. And from this observation, we can say, we can know the concentration of the band B microorganism in the compost from the blue color emission, light intensity of the blue color. And I don't show you here today, but so we already succeeded to design the probe for the band C microorganism. So our next step is to construct the DNA sensor. Okay, so I'd like to summarize my talk. The plant growth assay, this is the most reliable method, but it requires very long time, 6 weeks at least. Our DNA-based method, just one day is enough. So we can shorten the time considerably. This is the last slide. And we'd like to introduce the new idea of quality control when we produce a compost by using the DNA engineering and by using our new method, DNA-based method, to determine the compost quality. So we can introduce a new idea of quality control in the production of the compost. This is, bottom of this slide shows the motto of our laboratory, produce agricultural products as if they were industrial products. That's all for my presentation. Thank you for your attention.
Thank you very much. Any questions from audience? Yes. Would the consortia in the compost be specific to each type of waste used for making a compost? So using different waste would result in different group of microbes.
So that is a very good question. We confirmed so we can use this method for many types of organic waste, except the wood chip and sawdust and rice straw and other very hardly degradable organic materials. So the other organic materials such as sludge, the garbage, and food waste, and many other types of waste material, we can apply this method.
Okay. And the next question, in order to do compost of garbage, how
Yes.
are the temperature condition
Yes.
and
Yes.
moisture content? Could this factor effect on microbial community and organic matter conversion time?
Yes, that's true. So we already found out the optimum condition of the composting when we want to accelerate the most the composting. The temperature is 60 degrees Celsius. The moisture content should be in the range between 40 and 60%. And other aeration conditions and the pH and the other operational conditions we are already optimized.
Okay, one more question, but it is almost the same with the first question, I suppose.
Yes.
And it's one question from my side. Are those microbes are naturally occurred?
Yes, that's a very, very good question. So if we can use these two types of microorganisms as a seeding. At the start of the composting, we inoculate from the outside. And these two indicator microorganisms appear like a time bomb. So we can judge the good timing for the use of the compost in the farmland.
Okay, thank you, Professor.
Yeah, thank you very much.
Okay, the last speaker is Professor Michikazu Hara.
Hi.
He is from Institute of Innovative Research, and today's title is Sugar Conversion to a Monomer for High-Performance Polyamides. Professor Hara, please.
Yeah. Okay, can you see the slide?
Yes, we can.
Yeah. Sawadee Krab from Stu Hara, NDT Dial Jagdam. Good afternoon, ladies and gentlemen. I am Michikazu Hara, Laboratory for Materials and Structures, Tokyo Institute of Technology. First, I should like to take this opportunity to thank the organizing committee for giving me a chance of oral presentation. Today, I would like to talk to you about sugar conversion into an aromatic diamine compound. Biomass conversion has been extensively investigated to provide a renewable feedstock for the production of useful chemicals and fuels. 5-hydroxymethylfurfural HMF is an attractive intermediate for such a biomass-derived chemical platform because it can be further converted into various polymers such as polyesters and polyamides. For example, HMF is readily oxidized into 2,5-furan decarboxylic acid FDCA, a monomer for polyethylene flavonoate PEF. If we can replace PET by PEF, we can decrease carbon dioxide emission corresponding to 0.8%. In addition, HMF can be converted into 2,5-bis-aminomethylfuran AMF, a monomer for very expensive polyamides. However, this is nothing but building a castle in the air. We have no efficient processes for glucose conversion into HMF and HMF conversion into AMF. So, we have been trying to make catalytic systems available for both processes. First, I talk about HMF production from glucose. This slide shows the reaction mechanism for HMF production from glucose. Glucose is converted into HMF through isomerization, followed by intramolecular dehydration. First, we have to select promising catalyst candidates for glucose conversion into HMF. Boracic acid provides protons and act as catalyst for both isomerization and dehydration. However, boracic acid ain't effective for isomerization at low temperatures and can cause various side reactions at higher temperatures. While Lewis acid as electron acceptors can catalyze both reaction and milder conditions, but they also promote intermolecular side reactions, including aldol condensation, which result in the conversion of a large part of saccharides into worthless complex polymers. In addition, most Lewis acid can't work in water. Here, we have focused on anatase titania as a heterogeneous Lewis acid catalyst. Previously, we found that Niobium 5 plus unsaturated coordination spheres such as niobium-O4 tetrahedral on niobia act as Lewis acids, even in the presence of water, and the Lewis acid sites on titania, zirconia, and tantalum also act in water, as well as those of niobia. In groups 4 and 5 transition metal oxides, titania is the most attractive catalyst because titania is a ubiquitous, inexpensive, and stable solid oxide. So we use titania catalyst for HMF production from glucose. However, HMF yield and selectivity were only 30%. I mean, most glucose was converted into worthless complex polymers through intermolecular dehydration. Next, we phosphorylated titania surface to control the reactivity of the Lewis acid sites by the reaction between neutral OH groups on titania surface and blessed acid groups of phosphoric acid. Indeed, HM yield and selectivity over phosphorylated titania reached 56%, suggesting phosphate species on titania prevent glucose absorption, decreasing intermolecular dehydration. This improved the catalytic performance. However, this yield is too small to realize commercial HMF production. So, in order to improve the HMF production process, we have done mechanistic study to clarify the side reactions, and we found produced HMF to react with glucose on the Lewis acid sites. If we can remove and recover produced HMF from the reaction system immediately, we may decrease the side reactions. For this reason, we tried a biphasic reaction system composed of a water-insoluble organic phase and a water phase containing catalyst and glucose. In this system, HMF produced in the water phase is immediately extracted by the organic phase, preventing the side reaction between HMF and glucose on the Lewis acid sites on titania. Actually, HMF selectivity exceeded 80%, but the reaction system still requires improvement. This slide shows a bench-scale plant of the improved biphasic reaction system. You can see the produced HMF in a 1-liter beaker. In the improved reaction system, the isolated HMF yield reached 95% and the purity exceeded 99%. Of course, the catalytic activity didn't decrease even after 35 reuses. We have just estimated the HMF production cost, composed of capital and running cost, to be below $1 per 1 kilogram HMF. Thus, we got an efficient process to convert glucose into HMF. So, we began to make a route to AMF production from HMF. However, this isn't so easy. AMF yield obtained by a catalytic system based on a conventional reaction pathway is below 50%. No one wants to use such a reaction system. This shows a proposed route for AMF production from HMF. In this route, hydroxymethyl groups in HMF are oxidized into hormyl groups, forming dihormylfuran DFF. In theory, the reductive amination of DFF easily results in AMF. However, produced AMF readily reacts with DFF, forming this dimer. And amino groups of the dimer readily react with hormyl groups of DFF, forming trimer. such a reaction successfully proceeds, resulting in worthless polymerized compound. For this reason, we gave up this route via DFF and focused on a new route. I mean, first, the polymer groups of HMF are converted into amino groups through reductive amination. Next, alcohol of the resulting aminomethyl hydroxymethyl furan, AHF, is directly converted into amino groups by using borrowing hydrogen strategy. To realize this route, we have to fix two subjects. The first subject is the reductive amination of polymer groups bonded to aromatic compounds. Reductive amination is a familiar method for amine production in organic chemistry. In Japan, undergraduate students learn this classical method at the early stage of their universities. But the reductive amination of aromatic hormonal compound isn't so easy. This slide shows aminomethyl furan production from far by reductive amination in the presence of heterogeneous catalysts. As you can see, a conventional catalyst can give a product yield more than 99%. This is natural. Conventional catalysts can't prevent the hydrogenation of aromatic rings. This is computational free energy diagrams of the reductive amination of far and related side reactions. From the point of view of thermodynamics, it's really difficult to produce only target product. The hydrogenation of furan ring is favorable for the reaction system. This is also supported from mechanism and material science. Many transition metals have electron donation capability, and electron donation from transition metal d orbitals to antibonding pi orbitals of aromatic rings weakens pi bond. These pi bonds are easy target for hydrogen atoms on transition metal surfaces, resulting in the hydrogenation of aromatic rings. In order to catalyze reductive amination, preventing the hydrogenation of aromatic rings, we have to use an artifice. As mentioned before, transition metal particles have electron donation capability. If we can weaken the capability, we may get catalysts suitable for our purpose. By the way, there are Lewis acid sites on groups 4 and 5 transition metal oxides such as titania and niobium. Indeed, I showed the Lewis acid sites on titania converting glucose into HMF. Because electrons run short on Lewis acid sites, these oxides are electrophilic. As a result, depositing transition metal particles on these groups 4 and 5 transition metal oxides is expected to weaken the electron donating capability of the transition metal particles. As you know, any intermediate toward amino groups are subject to hydrogenation, compared with pi bonds of aromatic rings. So only hydrogenation of any intermediate may proceed on such transition metal catalyst. I think this is the opposite end of ammonia synthesis catalyst. We have been reporting strong electron donating to transition metal particles considerably to enhance ammonia formation rate. This slide shows the reductive amination of far, a test reaction in the presence of the catalysts. Rhodium alumina catalyst, which is the most active hydrogenation catalyst, can't prevent hydrogenation of furan rings. As a result, the yield of target product was only 60%. While ruthenium silica catalyst has a better catalytic performance than rhodium aluminum catalyst, but the target product is further hydrogenated on the catalyst. On the other hand, ruthenium niobia catalyst, ruthenium particles deposited electrophilic niobia, exactly prevent the hydrogenation of furan ring, and the product yield exceeded 99%. We used the catalyst for AHF production from HMF and found the isolated yield of AHF to reach 96%. This catalyst may be applicable to various reductive amination. The result for the reductive amination of various reactants over ruthenium niobia catalyst are summarized in this table. As you can see, this catalyst acts as an effective catalyst for various reactants. Please see entry 5. The target molecule is produced without the removal of bromine. Apparently, this catalyst is available for various reactants. In order to clarify whether the catalyst of ruthenium niobia is due to decrease in electron donating capability or not, carbon monoxide and soft catalysts were examined through FTIR measurements. Increase in electron donating capability of ruthenium readily increases electron donation to antibonding pi orbitals of carbon monoxide, weakening CO bond. Here, carbon monoxide acts as a probe molecule. For example, in the case of ruthenium silica catalyst, CO stretching band of CO bonded ruthenium appear at 2012 wave number. On the other hand, the stretching band in the spectrum for ruthenium niobia catalyst is observed at 2026 wave number. This means that ruthenium on niobia has weaker electron donating capability than that on silica. So weak electron donation results in the high catalytic performance of ruthenium niobia. Next, I talk about AMA production from AHF through borrowing hydrogen. Borrowing hydrogen is a new concept strategy applicable to direct amine synthesis from alcohol. In borrowing hydrogen, the hydrogenation of alcohol forms hormone groups. The resulting hydrogen atoms are used for the reductive amination of hormone groups. Borrowing hydrogen has a higher atom efficiency, doesn't need hydrogen molecules, and proceed in one reactor. So we try to synthesize AMF from AHF by using borrowing hydrogen strategy. Actually, a homogeneous ruthenium complex catalyst gave a high yield of AMF. However, I don't like this catalytic system for the following two reasons. First, this is a homogeneous catalytic system requiring a large energy consumption for the separation from the product. Second, the homogeneous catalysts exhibit high catalytic performance only in alcohol solvents, which is different from toluene, an effective solvent for first-step reductive amination. In addition, this is very expensive. For this reason, we developed a new heterogeneous catalyst effective for borrowing hydrogen, that titania-supported ruthenium magnesia catalyst. In the catalyst, metallic ruthenium and magnesium nanoparticles are fixed on titanium support. In the catalytic system, magnesia doesn't only accelerate the hydrogenation of alcohol by ruthenium, but also enhance the reactivity of hydride anions by electron donation from magnesia to ruthenium particles. This shows the FTIR spectra for 2-propanol adsorbed conventional ruthenium titania and our ruthenium magnesia titania catalyst. In the case of conventional ruthenium titania catalyst, the bond due to hydride anions appears at about 2000 wave number. On the other hand, in the case of ruthenium magnesia titania catalyst, it was observed at 1950 wave number, which is much smaller than 2000 wave number. This suggests hydride anions on our new catalyst has much higher reactivity than those of the conventional ruthenium titania catalyst. Indeed, ruthenium titania catalyst doesn't work for borrowing hydrogen, but ruthenium magnesia titania catalyst gave a high AMF yield exceeding 90% even in toluene. Of course, we confirmed these catalysts are available for various reactants as shown in this slide. To summarize the foregoing result, we have developed new catalytic processes for the production of HMF and AMF. Now, many people are working on commercializing these processes. We hope that our catalytic systems contribute to indispensable polymer production from biomass. Kopken Samrap Gan Lap Van Wani Krap.
That's all. Thank you very much. Any questions from audience? It's one question. To produce HMF from glucose, how pure does the glucose have to be? The purity of the glucose.
Yeah, that's a very good question. So there is no meaning in glucose concentrations because separation purify organic phase separation purifies HMF purity. So glucose concentration has no mean. Okay.
Okay, so today you mentioned from glucose to HMF and to AMF. And how about the conversion from biomass to glucose? Do you have any idea about this step?
You mean the...
What kind of biomass are you thinking of? For example?
I mean, so pentose, as you know, pentose, such as xylose, is converted into flour. On the other hand, glucose, aldohexose, such as glucose, are converted into HMF. So sugar is converted, all sugar is converted into furan compound. And yield, each yield exceeds 95%. Okay.
Yeah. And yeah, generally speaking, one of the key issues for biorefinery is cost, I suppose. And in your presentation, you achieved high yield with a good catalyst, good cheap catalyst. And how about the cost of the product? Is that
okay? So as mentioned before, the production cost of HMF is below $1 per kilogram. And AMF production cost is about $5 per kilogram.
$5 from HMF or from glucose?
Yeah, from glucose.
From glucose.
Yeah.
So maybe one of the issues is the price of glucose.
So, as you know, $0.4 per kilogram, 40 cents per kilogram. But that is a worldwide market price. As you know, in Japan, glucose is very expensive, exceeding $1. So we can't use glucose in Japan. So another country, we need another country.
I see. And I think your method is useful to convert some aromatic compounds from plants.
Yeah.
For example, how about lignin? Of course, it's a kind of mixture, not a pure chemical, but
yeah. But as you know, lignin content is below 30% of biomass, and lignin is composed of, as mentioned before, so many aromatic compounds. Such compound isn't available for chemical production. I mean the material production from lignin is not so large.
Okay, any other question from audience? Okay, one question. Have you ever tried to transform HMF to FDCA?
Yeah, it's very easy. HMF is readily converted into FDCA in the presence of manganese dioxide. And it's simple oxidation reactions. Okay.
Yeah. Any other question? Okay, there might not be other question. Thank you, Professor Hara.
Thank you. Thank you very much.
Okay, it's almost time to close this event. And if you have any more questions, please use questionnaire. As I mentioned, please cooperate with the questionnaire. We would appreciate your frank opinions, and today's presentation materials will be made available to those who complete the questionnaire. And should you wish to collaborate with us with Tokyo Tech, please feel free to contact Tokyo Tech ANNEX Bangkok. Okay, so all the speakers for today, could you show your face again? I'd like to thank all the speakers who gave us good topics today, very interesting topics today. Thank you very much. And Tokyo Tech Research Showcase is an annual event, and we are planning the next showcase for 2021. Thank you again for joining us today. See you next year. A Kop Makrap, Daupop Gammaina Kap. Bye-bye.